Hello and welcome to another episode of Plant Gene Podcast. My name is Ved Prakash and I am a postdoctoral fellow at Department of Plant Pathology, Kansas State University in Manhattan, Kansas. The Plant Gene Project centers around the enhancement of plant genetic engineering to ensure a sustainable supply of food, feed, and fiber. In each podcast episode, we welcome esteemed leaders from academia or industry to share their experiences and insight for the benefit of our listeners. Today, our guest is Dr. Inez Slamet-Loedin from International Rice Research Institute, IRRI, located in Philippines. Inez is currently the head of the Rice Genetic Design and Validation Unit at IRRI and is assigned as the global lead of the CGIAR Gene Editing Initiative. Inez is also a member of the Plant Gene Advisory Committee. Her research portfolio includes improving nutrition through healthier rice, disease resistance, drought, low phosphorus tolerance, and rice productivity. Inez is also an elected fellow of the World Academy of Sciences for the advancement of science in developing countries. She has published articles in reputed journals such as Nature, Nature Biotechnology, Nature Plant, Nature Communication, Nature Genetics, Proceedings of National Academy of Sciences, Plant Biotechnology, and many more. Welcome, Inez, and thank you for joining us today.
Thank you for giving me this opportunity.
Yeah. So let's start with the basics. Can you describe what a day in your job is like?
Right. My days really varies. I think sometimes I have to go, of course, mostly to my office. Sometimes I have to travel or I work from home. So generally, maybe I woke up, did 5 minutes, do 5 minutes quiet time, and then I have to travel about 1 hour to my institutes because I live in between my work and my daughter's workplace, and so I usually goes to screen house first. I go then to check the plans and everything, and then to the lab, and then have a couple of planning meetings, check emails, check labs, greenhouses, and work afternoon. I work on either I go to the lab and checking things and discussing stuff, or work on project proposal and have a more problem solving meetings. And when I go home, I have twin daughters, one with extra chromosomes. So, but she works in a school of all abilities. And the other one is just completed her master in oncology in Oxford. And so when I went home, we chat, we laugh and tease each other actually. And then in evening, because I have the collaborators globally, so sometimes I have also meetings is inevitable, I think. And then sometimes I travel, I think every 2 months or every month to different part of the world, to collaborators or to the either in the advanced countries or on our collaborating with the national partners. I think that's more or less my basic day.
That's
Thank
nice.
you.
Great. Can you describe your research?
I have two major works, both in on genome engineering. The work that we started about more 10 years ago or 11 years ago is basically aimed to making rice healthier with to increase iron and zinc in the milled rice, which is mostly rice is mostly consumed milled. I think I'm sure that you are very used to it too. And we use one gene from rice, one gene from apple to and the rice gene is the transporter basically, and because if you look at rice, there's a lot of iron in the leaves, but it is not transported to the grain. So, and also the gene to basically to bind the iron in the grain. And so now we have already at the stage of, we finished the multi-location trials in the Philippines and in Bangladesh for that. And then the idea is to combine it with golden rice. Depends on what is the market need, or we go ahead with the iron and the zinc rice. And I also work on photosynthesis to basically aim to improve yield with the collaborator from Oxford and Minnesota, and also with the EU consortiums with different approach. One is with the transcription factor, basically, and the other one is more multi-gene approach. And I also work on disease resistance, and this is with the partner from multi-partner as well from EU and from India and also from US. And another disease resistant project, which is very exciting, which is basically based on prime editing, that is with Cesaro, Australia. We also do surface for gene function validation. So like a breeder have a candidate genes from either their GWAS work or other works, and we do like knockout and overexpression, and sometimes they develop it later using marker technology. That's cover more or less, and both in traits towards geared to climate change and also healthier rice.
You mentioned about a gene from rice and a gene from apple. So, do you mean you are trying to incorporate a gene from apple into rice, or?
That part is the transgenic. Okay. Okay. So when we cannot do gene editing nowadays, we do more and more gene editing. But when we tried, but with at the moment, what we tried with the gene editing, we got the increase, but we also got the increase on the yield, which we tweaked the promoter to increase the expression. But with this insertion of rice gene with the different promoters, and also basically iron binding protein from apples. It was initially we work on soybean, but then we have also alternative. We try apple and beans. I think apples, it's also work well. So those things. So, but that has to go through all regulatory process. And so we work both on the developing the data for regulatory science and also but we work closely with the national partners for both.
Very nice. Well, another question I have for you is, what motivated you to pursue a career in science?
Oh, I love science. I think mainly because the innovative part of it, it's very exciting, but every day, even if it's a small things, you said, what have I now? You discuss with your staff, and then you look, and then if there's something doesn't work, then you have to tweak here and there, problem solving. I think it's like I have been asked a couple of times to work 100% on administration, but I eventually turn it down, consider it. But I mean, I have to do some, of course, everybody has to do some admin work, but it doesn't, if it's 100%, I don't think it's not me, because I love the discussion with other people, the challenge and the innovative part of it, I guess.
Very nice. Very
Yeah.
nice. Okay, so why are you interested in rice?
Rice is life. It's feed more than half of the global population, mainly in this part of the world, but it's actually now even in Africa now, they start to also consume rice. But on the other hand, of course, you don't want people to rely only on rice. So diversity is important. But then I found also it is like, at least if you can improve the rice with nutrition, more nutritive rice, you give the basic needs. No? But unfortunately, a lot of people, a lot of population that consume rice, it's also those in the countries on the transition or developing countries. So they don't always have an access to a good diverse diet. So I think that's why I was like, if you can improve rice, probably you can improve a little bit of the welfare of the people. And I think another thing that I like, there's the say in Indonesia is rice cultivates nature of humility. The more it have grains in its plants, the more it bows. So I think, but yes, so this combination that you can, if you can improve a rice-based agri-food system, you probably can improve poverty. You can do some poverty alleviation. Your contribution may be small, but you feel good because you do something.
I agree with you on that. So, what questions lately have you found interesting while working with rice?
I think the most interesting that I found is last 1 year is we work with our partners on Australia, just as IRRI on they found in barley and other cereal that if you change this allele and the difference is only one base pair, and then they found it's give a lot of improvement on the fungal resistance. And we first tried the transgenic one, and it did help on the it did improve the resistance. So, and then I thought, oh, but because we specialize on indica rice, and indica rice is more recalcitrant in tissue culture, right, in compared to japonica. So I thought, what is the percentage of editing that we're gonna prime? Because in this case, we have to do prime editing because of the base pair that we want to change. And we look at the publications, so we did all this tweak here and there. And I was quite, first, my PhD student got 50 to 80% replacement. I thought, oh, you must make a mistake, maybe double check on this. And then it's true. So I thought if we can do allelic replacement, it's a really game changer in the editing world, because that means that you can more and more use information of the available from land races and others. So I was very excited about it. And also the fact that's one part. And the other things is, as I mentioned, that we work on improving iron and zinc. So my colleague in Cornell University, he tested, we send the grain and they tested on chicken feeding assay, and improve the microbiome. So that's another side that, okay, first I also learned how if you are really between 1 and 5 years and lack of zinc, they show how the brain development of people who have sufficient, it's not stunted, zinc deficient, zinc deficient it's one of the major cause of the stunting, and you get a lot more complex brain development if you have sufficient, and it's irreversible. And this another thing, when we test it in the field, we have no yield penalty, and the zinc that we got is perhaps is quite much higher, from 60 to become 40 ppm, while the breeding can only do until 24 ppm in the grain. So, and then at the same time, it can improve microbiome because the microbiome is big now, right? And it's actually affecting a lot of things. So that is also two things interest me recently. The prime editing and also the chances to make rice healthier. And also I work with a collaborator who who in IRRI, Dr. Nese, that to reduce the glycemic index of rice.
Very interesting.
Okay.
So what role do advanced biotechnologies such as gene editing or marker assisted selection play in your research to develop improved rice varieties?
I think it has a very important role for gene editing, and even not necessarily as to make a final product, even for the, like, for example, the development of low glycemic index rice. They have a candidate genes, we check, and it does together, we check, and it's actually the gene responsible for it. So they can use it as a gene-based marker, which really accelerated and more accurate. And that is one thing, like for functional genomics part. But at the same time, I mentioned about, for example, the collaboration we have with Dr. Wolframer from HHU and Bin Young and other partner there that we, in this case, that Bin Young in doing the gene construct, but we do the editing and the selection of it. And then it was checked in Colombia, and in both US and Colombia, they make it, they got status for non-regulated status. And now in the next phase of the project, even we are not involved, but it is in the next phase, it would be more than our partner. So ICAR from India will further test it, which is, and that's basically edit the promoter's region. So the tal effector from the bacteria could no longer bind to it. And also we worked before with Tungro at IRRI, and also we basically edit the place that the sequence where the virus bind, and then which needed for translation initiation factor. And that is also, I mean, all this clearly has the role to have an improved rice varieties. And I think initially perhaps takes longer, but it is more and more efficient. But this can only be done through partnership, because both partner in Advanced Research Institute, but it's also in the national partners. And I think really best if you involve everyone at the very beginning, so they have a sense of belonging. This is our projects together. And because at the end, if there's no adoption, it's become a publication only. But if it's adopted, and it's and you really have to work with the advanced cultivars for that purpose, and depends on the purpose. So I think it does play a it will be more and more when the regulation is more towards balanced approach. But I think if you look even the map now, initially only few countries regarded gene editing as the breeding lines, but now it's more and more you see countries. Even in the last 2-3 months, things are moving so fast now. So I really have a big hope on. I mean, it is not silver bullet. We also always to realize that this is one tool of the breeding. So, but it is a very potential tool.
I have to appreciate that you are working in so many different aspects, including resistant with virus, resistant with bacteria, resistant with fungi, and drought, which is abiotic stress. So that's very impressive.
Yes. Drought is very challenging. Actually, I work on transgenic before on drought, but for gene editing, because drought is a complex trait, it's still challenging. I think those are the set of the challenge that we need to have in it. But we do work on, but we can do editing directly on the lines which is already have a partial drought resistance, for sure.
Sure, sure. Okay. So another question I have for you is that scientists have been going back to the wild relatives for incorporating various beneficial traits into cultivated varieties. What is your thought on this?
I think it is one of very important aspect on this on the potential of the technology. You mentioned that currently I'm responsible for uh, CGIR. CGIR is a consultative group on agriculture, and it covers many crops, right? Uh, so currently there are 5 centers who already work on genome editing, which apart from rice, there is a banana, cassava, sorghum, maize, and potato would start soon. So those are among that potential and crops that we have been working on. But if I look at the CGR gene bank collection is recognized among the best in collection gene bank in the world. But despite of major investment that on the area of genetics, genomics, which a lot of sequence are now available, right? Potential genetic pathway, but actually in the breeding mainly use the core collections. I think because of the linkage drag and all the unknown of the land races, so it's not being used so much. So I think this development of gene editing technology and available of large-scale sequence data and phenotypic studies would be very important to break the yield barrier, because like in rice we have the minor increase now for like we probably got 1%. It's already very, very good if we got annually increase. It's not like maybe corn or something, but if we can get more and more genes from a landrace and wild relatives, I think it would potentially you break the yield barrier, and gene editing is very useful to speed up the breeding process in this way. And it also can be used to shuffle genetic information for in the pre-breeding generation. If you stimulate the recombination, for example, improve the recombination, so transfer of the exotic information from cultivated from this is possible from gene. And also, of course, the domestication, like Rodwing has tried to do also. I think I would be also next if we can do, because we have a lot of wild rice collection and you don't know, many genes unknown. So it's either for functional validation or domestication is a big avenue. But for Teresia quartata, for example, it's really salinity tolerance that you can grow it in the salt water. So for the climate change, I think it is super important to adaptation on the climate change. And it probably also can do some mitigation things like to reduce nitrogen use or methane production. Yeah. So I totally agree with you that the wild rice relatives, I would be gene editing can have a big role on getting some important allele from that, and also to get more and more knowledge information.
Yeah, completely agree. And in fact, I am also screening wild relatives from wheat. and screening and
Ah,
tolerance against virus. So I got
excellent.
interested into what is happening in rice.
That's very exciting work.
Yeah, it is. Okay, so let's move to the next question. So, how do you collaborate with other researchers, agriculture organizations, and farmers to ensure that your research findings are effectively translated into practical solutions?
I think, as I mentioned, CGIR, basically most of the CGIR center, uh, we are like an honest broker. We work in the middle between advanced research institutes and the national partners. As I mentioned before, the concept of co-development is very important. It's not, in the past, we tend to develop something, and in a certain scheme, we still do that. Then we develop something, we share after we develop something. But it's also very good if you can co-develop together. So we work with the Advanced Research Institute for the most advanced technology to apply it for, because mostly they work with the model species, right? So we apply it in the elite germplasm because we have very good transformation facility or capacity for indica rice. And basically we can transform any indica rice with different frequency, but we have that capacity. But it's very important to link with the both national agriculture and extension system and then partnership. But we also have a partnership with private sector. It's either small, medium enterprise or big private sector which has the access on the technology. And another point that it's very important to eventually able to share and get it's adopted to actually work on the intellectual freedom to operate for intellectual property part. And for that, you need to, depends on what is your product. If your product can, you can negotiate for humanitarian purpose. So we work supported by a diverse network, and you also need to discuss with investor because a lot of our project is supported, for example, by Bill and Melinda Gates Foundations, by UK FCDO, by ACR from Australia, and USAID. And so all these are it's quite complex things, but developing network is very important. While you are also need to work very well with the national partners. I think to really treat it everybody equal also very important. Thank
Absolutely.
you.
Yeah. Okay, so what advice would you give to students or researchers interested in pursuing their career in your field?
I think important to have passion, perseverance, because experiment would fail from time to time. And then you need to have a vision, what you want to do in future. And while you are you're attending conference or things, you really need to develop network. So sometimes you tend to go and stick with your friends only. Don't do that, because it doesn't mean you don't care about, but that's an opportunity. If you go to the conference, you open network, because many people are open would like to share their knowledge and information. But passion is important, and also do not scare for change. I moved from Indonesia to here, and it make a big change on my career. Moved to other places. I mean, it's difficult at the beginning, but it is important, I think.
Absolutely.
I think those are a few things that I think
Okay.
but passion is also number one.
Okay. I have a lot of questions, but time is less, so maybe I will move to the last question I have. So, what
Yes.
impact do you hope plant gene will have on transformation?
I think the fact that it has ideas to address global demand for plant transformation and gene editing through network is very important, because more and more partnership, you can work on your problem solving, and also access on technology, access on even sometimes facilities, and if you need to do part of your works, and also probably in the future have also avenue to negotiate on the access, freedom to operate on the technology. I it has a very good mission and ideas, because initially people think that transformation is something uh, very easy to be, but with the gene editing, it's one of a game changer if you have a very good transformation system, including transforming wild rice, for example. Yes, and I totally support it, of course, with this I think very good initiative.
Great.
And that's hopefully, of course, I hope that it has an impact. Eventually, the product is adopted, and also more and more countries are open with to treat it as breeding lines, because it's basically a mutation
Right.
for breeding. By the correct communication and outreach, I think it's open a lot of avenue and opportunities.
Right.
So eventually your work, my work will get adopted and be useful.
That's true. That's true. Okay. So before we wrap up, I want to extend a huge thank to Dr. Inez Slamet-Loedin for joining us today and providing such a valuable information about their groundbreaking research in improving rice nutrition and agronomic trade. Your passion for your work and dedication to addressing global food security challenges is truly inspiring. We are incredibly grateful for your time and expertise. And to our listeners, thank you for tuning in. Stay tuned for more enlightening discussions in future episodes. Until next time, I would say take care and keep exploring the wonders of science.
Thank you for the opportunity, and all the best to you and all young scientists who listen to this discussion. Thank you so much for the opportunity.
Thank you. Thank you.
All the best. Okay.
Plant Gene would like to thank the National Science Foundation for their support. Plant Gene is funded through NSF grant IOS 2210962. If you would like to learn more about Plant Gene or become a member, please visit plantgene.sivb.org.